Hello everyone. We are starting with our live marathon session, which is going to be the practice of June 2023 previous year question paper. The last attempt of CSIR NET. So we will be practicing some of the PYQ from the part B. Okay, and it is going to be the morning shift questions. Join the session fast. We'll wait for few students to join and then we'll start the session. Good evening everyone. Good evening Akanksha. Share these sessions as much as possible because these are very fruitful and free live classes for you all, which are happening at Botany Insider at sharp 8 p.m. Okay. There are 7 students in the class already. Join fast. Good evening, Siva. How's your preparation going, everyone? I hope it is going fine because exam is near. Is my voice coming fine? Am I audible properly? Please confirm. Yes. Am I audible properly to you all? Okay. So first thing first, that we have a full course and a crash course running for all of you who are preparing for December 2023 exam. Okay, so if you are preparing for these exam, you can join in either of the batch. And for your full-fledged preparation, we have our notes booklet at Botany Insider app. Yes, we have a Botany Insider application that you can download from Play Store and iOS as well. Okay. Good evening, Shivani. Good evening, Kamla. Okay. So we are practicing PYQs today, the last attempt of the CSIR, which was the joint attempt of December 2022 and June 2023. We will be solving some of the PYQs here of the part B section, and these questions are from morning shift. Okay. Also, we will soon be launching our PYQ course. So if you want to practice all the questions from 2023 to 2013, all the last 10-year questions, so you can make sure that you are joining our PYQ course, which will be launching on Botany Insider application. And for all the updates of the class, we have a WhatsApp community group which you can join. The link will be provided in the description box. Here you will get all the updates of the coming courses and all the live marathon classes as well. Okay. So this is the first question. It is from the transport across nucleus from cell biology. Okay. So make sure that you are reading the question very carefully and then marking the option. Attempt this question. Everyone, I'm giving you 2 minutes max to attempt this question. Then we'll move forward. In the context of protein import in the nucleus, which molecule is responsible for releasing the cargo from the importing receptor? Okay. Good evening, Lena. Good evening. Read the question very carefully and then answer. And make sure you are joining daily at sharp 8 p.m. for the live marathon classes. Okay, very good. Others mark the answer. There are 16 students in the class. I want all of you to mark the answer. It is a very easy question. Okay, very good. Six students have marked the answer. Other 10 students mark the answer quickly. Yes, all of you got the answer correct. The answer is RAN molecule. So what happened in the nucleus import is that here we have, suppose this is our cell membrane. Okay. Here we have cytoplasm and here we have nucleus. So what happens is we have important molecule here which is going to import our cargo protein. It is going to bind to the import molecule and that is going to pass from a nuclear pore complex. We know that nucleus membrane has nuclear pore complex. So the cargo and important are going to bind to each other and these are going to pass through the nuclear pore complex and before releasing the RAN GTP is used up in this process, okay, and it is going to release the cargo here and important molecule here. Similar to that, we have an exporting molecule which is going to transport our cargo from outside of the nucleus to inside of the nucleus, and here RAN GTP is going to be used, okay. Here we use RAN guanine GTP is activating protein which is going to convert the GTP to GAP, and here we have RAN guanine exchange factor because we need GTP, so it is going to convert GDP to GTP in the process, and the main molecule which is used is RAN. I hope this question is clear to you all. Let's move forward with our next question. Yes. Read the question very carefully. It is a very easy and a repeated question which has been asked in previous years a number of times. Okay, in the match the following ways also, sometimes in statement wise questions also. So it is saying which one of the following is the communicating junction linking adjacent cells in plants which permit small molecule to pass from cell to cell while blocking the passage of most large molecules. Okay, the options are adherent junction, gap junction, plasmodesmata, or hemidesmosome. Okay, everyone read the question very carefully. There are 20 students in the class. Make sure you are reading the question very carefully. And if you want to practice the PYQs in this manner for the last 10 years, you can join our course which is launching for the PYQ session itself on our application. Okay. Very good, very good. 5 students have marked the answer. Others quickly mark the answer. Everyone, yes, very good. It is asking specifically for plants, okay? Read the question very carefully. I want all the students to participate in the class. Everyone mark the answer. Yes, the answer is Plasmodesmata. Plasmodesmata is a type of gap junction which is present in plants. Okay. Gap junction also performs the same function, that it permits small molecules to pass from the cell while blocking the large molecules, but it is asking specifically for plants. So for plants, the answer is going to be Plasmodesmata. Okay, there are students who are marking gap junction in the hurry without reading the question. Okay, so make sure you won't repeat this mistake again. Okay. So plasmodesmatas are adjacent plant cells which communicate with each other through cytoplasmic connection, which are known as plasmodesmata. They function analogous to the animal cell gap junction. Okay. Plasmodesmata are intracellular channels that establish a symplastic communication pathway between neighboring cells. At each plasmodesmata, the plasma membrane of one cell is continuous with that of another. Okay? Yes. Read the question carefully and answer this. This is a very easy question from molecular biology. Okay? It is from the section translation. It is saying which one of the following translation factor is used as the step of translation initiation and defined as anti-association factor of 30 and 50s subunit. Yes. Read the question very carefully and then mark the answer. Okay, one student is already marked. Others? This is a very factual question, and these type of questions are the most important type of questions in CSIR because part B contain one-liner statements mostly. So make sure that you are checking your facts and revising properly. Everyone mark the answer fast. These initiation factors have been asked in match in the following also. Okay, the initiation factors and their associated functions. So make sure you are revising and recalling them. 5 students have already marked. Others, please mark the answer quickly. Yes, the answer is initiation factor 3. Okay, initiation factor 3 is the factor which is present in anti-association properties for 30s and 50s subunit. It prevents the 30s subunit to bind to 50s subunit before the initiation translation process completes. Okay. initiation factor binds to free 30th subunit and prevents premature reinitiation of the large and small subunit of the ribosome. And it has a second function in controlling the ability of 30th subunit to bind to mRNA. Also, it ensures the fidelity of initiation site selection, and it selects the initiator tRNA for use in initiation. So make sure you are writing all these functions of IF3 in your notebooks. Okay. Now read this question and mark the answer. Try to use the elimination method in this question. These type of questions are very analytical questions, and you don't require the prior knowledge. It is saying the extracellular domain of a cell surface receptor was switched with the extracellular domain of another receptor. So we have a cell membrane and there we have a receptor. So its extracellular domain is replaced with B. Okay. To create a chimeric receptor, rest of the part of the receptor are same, assuming that there is no effect on the functionality of the domain in chimeric receptor. What is the most likely outcome in the presence of ligand for the receptor B? Okay. Read the question, analyze the question, and then mark the answer. Don't be in a hurry to mark the answer. Yes, whether it could be the ligand will activate the pathway normally triggered by receptor A, or it will activate the pathway triggered by receptor B. The chimeric receptor failed to transduce any signal in response to the ligand, or the chimeric receptor will cause constitutive activation of the signaling pathway. Make sure in exam you are at least try to attend these type of questions, because they are very easy and you can get it right in one attempt only. Okay. Quickly mark the answers. We have 21 students in the class and I want all of you to participate in the class. These PYQs are utmost important. Quickly mark the answer, everyone. Okay, 4 students have already marked. Others, please mark the answer. Okay. Try to correlate this question with other signaling pathways that you have studied. What would have happened if you changed the receptor of one signaling pathway? Okay, there are different answers from you all. Quickly mark the answer. I'm giving you all 1 more minute to answer this question and then we'll solve this question, okay? Try to use the elimination method. That works best in these types of questions. Okay, only one student have got the answer right. The answer is option number 1. Okay, The ligand will activate the pathway normally triggered by receptor A, because if you read the question carefully, that it is only exchanging or switching the extracellular domain of the receptor, not the entire receptor. Okay, and it is saying that there is no effect on the functionality. The rest of the functionality of the receptor is going to be the same. So even if we switch the extracellular ligand, suppose we have a ligand of this shape and the previous A receptor was of this shape, and now we switched it with receptor B. Okay, it is going to bind to it and perform the same function that receptor A was going to perform. Okay, and there is no difference. It is written here very clearly that there is no effect on the functionality of the domains in the chimeric receptor. So the ligand will activate the pathway which is normally triggered by receptor A. I hope this question is clear to you all. For those of who you marked the answer wrong, yes. Now is the question clear? If you have any doubt, you can ask. Okay. Otherwise, we are moving forward with our next question. Others, confirm if the question is clear. Okay. Now read this question. It is based on the erythrocyte functioning. Okay. In mature erythrocyte, the end product of glycolysis, the glycolysis which is specially performed in RBCs. Okay. That contain the carbon of glucose is option A is ethanol, pyruvate, acetaldehyde or lactate. What is the end product of glycolysis which is present in mature erythrocytes? Yes. Try to recall the glycolysis cycle which is occurring in erythrocytes. And what is the end product of that glycolysis? Okay. Others, mark the answer. Very good. Others, quickly mark the answer. There are only 5 to 6 students who are participating in the class. I want everyone to participate. Okay, few students have marked the answer. Others, quickly mark the answer. Whether is it ethanol, pyruvate, acetaldehyde or lactate? What is the end product of glycolysis in mature erythrocytes? Try to read the question twice without answering. Answer is option number 4, lactate. Only one student has got the answer correct. It is lactate. Very good, Savita. So in mature erythrocyte, the end product is not pyruvate. It is the product of normal glycolysis. But in case of erythrocyte, there is a specialized glycolysis which happens, in which case the end product that forms is lactate. Okay. Mature erythrocytes are devoid of mitochondria and therefore they cannot undergo cellular respiration. So there is no cellular respiration that happens in case of mature erythrocytes because they do not have mitochondria. Instead, they rely on glycolysis to generate the energy. The end product of glycolysis in mature erythrocytes is lactate, which is then released into the bloodstream. Okay. Now is the question clear? For those of you who got the answer wrong, now is the question clear to you all? And make sure that you are covering the glycolysis in RBC. Okay. In mature RBC, there is specialized glycolysis because they are devoid of mitochondria. Now attempt this question. This is from ecology and this survivorship curve has been asked in CSIR multiple times. So make sure that you are covering this topic in the best manner possible. And if you want to cover this topic from Botany Insider, you can purchase our course, which is full course and crash course that are running. Okay. Read the question carefully. Which one of the following statement is correct for primary successional species? Okay. What type of survivorship curve do primary successional species follow? Make sure you are clearing out your facts because this is a very factual questions. Yes, answer quickly, everyone. Or even if you don't know the answer, just try to attempt the question. No worries if even if you got the answer wrong. There is still time for you to practice these questions. Okay. Others mark the answer. Yes, try to think of the characteristics of primary successional species and then apply your knowledge to survivorship curve species. Okay. Or even they do not follow species survivorship curve. Okay. Others mark the answer quickly. This is the very important topic of ecology. Also comment what are the primary successional species and their characteristics. Yes. What are the primary successional species? What is your understanding of primary successional species? Everyone mark the answer. Yes, the answer is option number 3. These species, no, option number 3, yeah. These species show type 3 survivorship curve. Okay. So what are primary successional species? These species are the type of species that colonizes the barren land. They are the first species to colonize any barren land or any lifeless habitat, okay, which do not have a life before. So they have characteristics like quick growth, they grow very rapidly, and they have high competitive advantages. So all the types of primary successional species are matching with type 3 survivorship curve. This is the last question for today. Make sure you are reading the question. This is also from ecology, from population diversity concept. Okay. Which is the best method suited to estimate the population of the size of fish? Is it camera trap, line transect, point count, or mark recapture? Yes. Quickly mark the answer. Which is the best suited method to estimate the population size of fish? Quickly, there are 17 students in the class. Everyone participate and mark the answer. Yes, very good, very good. Others, mark the answer. Yes, the answer is option number 4, which is mark recapture technique. This is the technique which is used to estimate the population size of the fish. Okay, these were all the questions for today, and we will do more questions in our PYQ sessions in the class, which will be launching very soon on Botany Insider application. So make sure you are enrolling in this course and practicing PYQ in the best and structured manner possible. Okay, also we have a full course and a crash course running. So if you feel any lag in your preparation for December 2023, you can join these two batches, and also you can purchase our notes booklet. Okay, it was a very good session and very interactive session. Was this session helpful to all of you? We will daily practice PYQs and other important topics of CSIR at sharp 8 p.m. So make sure you are joining these live marathon classes and sharing them as much as possible. Thank you everyone. Thank you so much. Thank you Siva. Thank you Kamla Kumari for joining the session. Bye bye everyone. Take care.